Lab 6. Bacteria in the environment. Introduction. Bacteria are often maligned as the causes of human and animal disease. However, certain bacteria, the actinomycetes, produce antibiotics such as streptomycin and nocarticin. Others live symbiotically in the guts of animals, including humans, or elsewhere in their bodies, or on the roots of certain plants, converting nitrogen into a usable form. Bacteria put the tang in yogurt and the sour in sourdough bread. Bacteria help to break down dead organic matter. Bacteria make up the base of the food web in many environments. Bacteria are of such immense importance because of their extreme flexibility, capacity for rapid growth, and rapid reproduction. Bacteria are so widespread that it is possible only to make the most general statements about their life history and ecology. They may be found on the tops of mountains, the bottom of the deepest oceans, in the guts of animals, and even in the frozen rocks and ice of Antarctica. One feature that has enabled them to spread so far and last for so long is their ability to go dormant for an extended period of time. Bacteria have a wide range of environmental and nutritive requirements. Most bacteria may be placed into one of three groups based on their response to gaseous oxygen. Aerobic bacteria thrive in the presence of oxygen and require it for their continued growth and existence. Other bacteria are anaerobic and cannot tolerate gaseous oxygen, such as those bacteria that live deep in underwater sediments or those that cause bacterial food poisoning. The third group is the facultative anaerobes, which prefer growing in the presence of oxygen but can continue to grow without it. Bacteria play important roles in the global ecosystem. The ecosystem, both on land and in the water, depends heavily upon the activity of bacteria. Bacteria are decomposers. Decomposition is the breakdown of these dead organisms and the conversion of organic molecules back into inorganic molecules. Inorganic molecules would quickly be depleted if not for the activity of decomposers. This may not sound too bad to you, but realize that organic molecules contain carbon. The carbon comes from the carbon dioxide that plants take in from the atmosphere. If decomposers did not return the carbon to the atmosphere, there would be no more carbon. Without carbon dioxide, there would be no photosynthesis in plants and no food. Procedure. Day 1. Obtain a TSA plate and write your partner's initials and your own on the top using a grease pencil. Number 2. Take a cotton swab and lightly moisten the tip with distilled water. Number 3. Swab a chosen surface such as your desk, the floor, a doorknob, or your shoe sole. Number 4. Quickly open the lid of the TSA plate. Gently touch the inoculated swab to the nutrient auger plate near the edge. Quickly close the lid. Number 5. Next, take an inoculation loop and streak the plate as shown in the illustration to disperse the bacteria over the rest of the TSA plate. Number 6. Tape the lid of the Petri dish. Do not open again. Number 7. Place upside down in the collection basket. Number 8. Record on your lab sheet the location where your bacteria sample was taken. Day 2. Do not open your Petri dish. Number 1. Retrieve your Petri dish from the collection basket. Number 2. Do not open your Petri dish. Observe the different colonies of bacteria. Look for different bacteria colonies based on the size, the color, and the colony shape. Record this data on your handout. Use the diagrams on the following page to describe the shape of the colonies. Number 3, make a detailed drawing of your Petri dish. Label the drawing and diagram the location of each of the different colonies.